Concurrent molecular characterization of sandflies and Leishmania parasites by amplicon-based next-generation sequencing. Three major forms of Leishmaniasis. Cutaneous Leishmaniasis, CL, caused by major tropica infantum or Mexicana brasiliensis chagasi. And mucocutaneous Leishmaniasis, MCL, caused by Brasiliensis mexicana and major or tropica. Visceral Leishmaniasis, Donovani infantum and Chagasi amazonensis. Infection components: human host, sandfly vector, parasite, and reservoir, such as rodents, rock hyrax, humans, or dogs, depending on the parasite species. Global distribution. In blue are the 10 countries where 85% of the CL cases are found. In green, the 4 countries where 90% of the MCL cases are found. While in red, the 9 countries where 95% of the VL cases are found. Objective: to develop a novel amplicon-based NGS for the simultaneous identification of individual sandfly species and the Leishmania infection within, in a bulk sample. Methods: sandfly collection and DNA extraction. Trapping of sandflies in Tubas and Al-Khalil during the biting season, May to September, using sticky and CDC traps. DNA was extracted by PCI method. DNA reference samples. Sandflies: Phlebotomus, the Old World, Sergenti perflievi seriacus babatazi tobbi and argentibus. Lotsomaya, a New World, Guianensis, Sergentomaya, Shvetsi, Leishmania, Tropica major infantum, Donovani and Ethiopica. Target genes and primers. The ribosomal RNA transcription unit, rDNA-owned, in both Leishmania and sandflies were targeted. For sandflies, 18S ribosomal RNA was the target gene, while for Leishmania, ITS1 was the target. Methods: single-plex PCR. Both PCRs, sandfly and Leishmania, were run separately to check for the validity of the system. Multiplex PCR, Leishmania and sandflies. Leishmania and sandfly in one tube in a total reaction volume of 25 macroliters. Note the long primers. The black regions are Illumina adapters, which should be the same for all forward primers and reverse primers. Only the colored sequences represent the original primers specific for each target. Standardization of multiplex PCR. Determine optimal concentration of sandfly primers and analytical sensitivity of Leishmania DNA by comparing results across a series of concentrations. DNA library preparation for NGS by two-stage PCR. The first PCR, primers are modified by adding Illumina overhang adapter to each primer to ligate flow cell adapters to our target fragment. In the second PCR, indexes or barcodes, 8 base pairs, are added to the primers as sample identifiers, in addition to Illumina adapters P5 and 7 to allow binding to flow cell. Virtual probe preparation for NGS. Sandfly species and Leishmania species were retrieved from gene bank and aligned in search of conserved variable regions, green rectangles, to select the probes that will be used to identify each species of the sandflies and Leishmania. Virtual probe preparation for NGS. The 15 sandfly species probes and the 7 Leishmania species probes have been fed into Galaxy software. Galaxy will be asked to give us the name of the species whenever it locates its corresponding probe. Statistical analysis. The statistical test, Spearman's correlation, Kappa coefficient, phylogenetic tree, multiple alignment, and primer design were conducted using these software packages. Sandfly identification by morphology or microscopy. Of the 187 collected sandflies, 9 species were identified. The vast majority, 76%, were Phlebotomus sergenti, the L. tropica victa, followed by Phlebotomus babatazi, the L. major victa. Results: multiplex PCR, optimal primer concentration for sandflies. 0.1 micromolar is the optimal concentration for sandflies, as it showed both bands without any dimers and strong Leishmania band despite low concentration. Results: multiplex PCR, feed collected sandflies. Here, the gel shows 18 sandflies at 230 base pairs, only 2 Leishmania bands at 343 base pairs, indicating 2 Leishmania infected sandflies. Analytical sensitivity. The table shows the minimum detectable number of reads of Leishmania as 493 reads, which corresponds to 0.0016 nanogram, 1.5 femtogram of Leishmania tropica. To confirm NGS, 2 full ITS1s were Sanger sequenced, which showed to be identical to NGS sequences. Results: NGS validation, 187 sandflies. NGS and morphology methods for the identification of sandfly species were strongly correlated by Spearman's correlation. Both perfectly agreed in 171 out of 187, indicating high reliability of NGS. Most of the disagreements were hard to distinguish by morphology due to high similarity between species. Results: phylogenetic analysis. The 18S ribosomal DNA neighbor joining tree showed 3 clusters based on genera: Phlebotomus, Sargentomyia, and Lutzomyia. This supports our NGS method, in which the 187 NGS sequence sandflies were arranged according to genera, without a single deviation. Discussion. 1. Novelty. First, one-step reaction to identify species across both sandfly and natural Leishmania infection from one wild-caught sandfly. 2. Robustness. Unlike conventional PCR, our method does not need extra confirmatory or genotyping methods. 3. Reliability. Sandfly identification by morphology and NGS were perfectly in agreement. NGS was supported by phylogenetic tree clustering. 4. Specificity. Our method correctly identified 5 Leishmania species and correctly distinguished 3 sandfly genera, as well as 6 phlebotomous species. 5. Specificity. Thousands of amplified sequences collapse into one consensus sequence, suppressing errors and increasing specificity. 6. Limit of detection, LOD, or sensitivity. LOD is 0.0016 nanogram or 1.6 femtogram, which is comparable to other highly sensitive methods. Conclusion. NGS is a powerful tool for the simultaneous identification of individual sandflies and Leishmania species. Useful for large-scale field studies because it's quicker, high throughput, valid and reliable, and cheaper per sample.